Vamos a esperar un poquito a que acaben de llegar las demás personas aquí al auditorio y que nos conectemos un par más de personas al Zoom y empezamos en unos 2 minutos. ¿Sale? Bueno, creo que vamos a ir comenzando. Steve, ¿quieres abrir tu cámara? Tal vez quieres dejar de compartir primero para que te veamos.
Aquí,
Ahí está Steve.
aquí
Bueno.
estoy.
Para mí es un muy gran gusto y un gran honor presentarles al profesor Steve Hoffman. Em, pues además de ser una persona muy reconocida en su área, es un gran amigo ya por muchos años. Steve tiene una formación de médico y en paralelo hizo también la formación de computólogo, entonces es médico y científico de la computación al mismo tiempo. También hizo el doctorado en medicina y luego hizo el doctorado también en ciencias de la computación. Y muy en la Universidad de Leipzig, que es donde yo lo conocí, tuvo su primer grupo junior, un grupo junior que se dedicaba pues a la creación de algoritmos para resolver problemas biológicos. En particular, estuvo contribuyendo muchísimo a la epigenética, a la metilación. Ha trabajado en muchísimos proyectos, sobre todo de salud humana, pero también con animales, con la hidra, regeneración y bueno, muchísimas cosas. Es muy interesante. Y desde hace más o menos unos 10 años, creo, está en el Instituto Leibniz para Envejecimiento en Jena, en Alemania, donde ya es pues profesor, como full profesor, y bueno, sigue esta línea de investigación. En su grupo se llama como el algoritmo del envejecimiento, ¿no? Entonces, pues estoy súper contenta de presentarlos. Estoy un poco no tan contenta de que no pudo estar aquí con nosotros. Él quería estar con nosotros aquí en persona, pero bueno, surgieron algunos imprevistos que no le permitieron viajar a México. Pero bueno, de todas maneras va a tener su presentación y pues, Steve, muchísimas gracias por aceptar de todas maneras dar esta presentación. Y adelante, ¿quieres hacerla en español?
No, por favor, no.
Pues español es muy bueno, pero Pero bueno,
Ah,
adelante. Entonces, esta presentación va a ser en
mentira.
inglés.
Okay. Yeah, Maribel, thank you very much for the very kind introduction, which is also a little bit rare to hear it from you. But in any case, thanks a lot for the invitation. Although I would have really loved to go to México, lindo y querido. Again, it was really not possible this time, but I hope to be there as soon as possible. All right. Without further ado, today I was told to also go a little bit into the direction of sequence alignments, specifically in the context of next generation sequencing. Now, what I'm going to present to you today is basically some research that is based on a specific tool, or let's say on a study that we conducted that led to the modification of existing tools that allow us to characterize the expression of transposable elements. And I have to say that the work that I'm presenting was mainly done by Robert, and Robert is a PhD, or has been a PhD student in my lab. He graduated last year, but he's still with us, and we are basically still working on this topic. So it's also for me a nice recapitulation where we are hopefully going in the future. Okay, so. So first of all, I want to say a little bit about the biology of transposons. Well, transposons, or the transposome, consists of transposable elements, and they can be found in basically all the genomes that we know. But there are some very interesting factoids about transposons. So what you can see here is the so-called peppered moth. And during the times of the industrialization, when large factories were built, this moth apparently experienced the insertion of a transposable element into its cortex gene, which led to another color. The moth became dark, so not this peppered original style, but it became dark. And because of the fumes that were exhausted from these large factories, this moth had an evolutionary advantage, namely, because of the color, it would not be found so much by so easily by its predators. So it turned out that this transposable elements really contributed to the fitness of the peppered moth, and it's one of the more prominent examples of what transposable elements have contributed to evolution. Specifically, when it's about color, you find all of these animals that have changed here the coat of the dogs, but also the color of fish, the fur of mice, and so forth, the color of eggs. So in all of these instances, we really see that transposable elements have, in this case, a visual impact on nature that we can perceive. And of course, the question is, what else can these transposable elements do? How do they shape evolution? How do they shape, in particular, gene regulation? So, as I said before, basically all the genomes that we know share to varying degrees um, transposable, or have transposable elements in their genomes. Humans, for instance, have roughly above half, roughly above half of the genome of humans consists of transposable elements. And other species, for instance, here the Japanese pufferfish, has a very low amount of transposable elements, whereas the lungfish, for instance, has a very high percentage of transposable elements in its genome. But as you can see already, it's basically present across all kingdoms and domains of life, whether you look at insects or plants or bacteria, you find in all of these instances these transposable repetitive elements in the genomes. When we talk about humans in particular, we find that transposable elements have been highlighted in the literature in the last couple of years quite prominently, like in this Cell paper, where Faulkner and his colleagues found that a family of transposable elements, here L1 line elements, um, were found to contribute to the mosaicism in hippocampal neurons, meaning that our brain, specifically the cells in our brains and our neurons, differ a little bit based on the insertions of L1 retrotransposon elements. So specifically, they shape not only the genome, but to some extent also what is expressed in these neurons. A later study then looked at transposable elements in mice, and it was found that these elements, these transposable elements, become active dependent on the maternal care that these mice received. And the activity of L1 elements, specifically in mice, results for some of these L1 elements into the reinsertion of these elements into the mice's genomes, meaning depending on the activity or depending on the maternal care, the L1 elements become active or not. And then these L1 elements are integrated into the genomes of the neurons of these mice. So, in essence, one could say that the maternal care in these mice changes the genome of the neurons, which is a quite striking result that has also caused some discussion. So I hope this is somewhat convincing that it would be really interesting to know more about the activity of transposable elements. And before I go on, I want to talk a little bit more about the biological consequences. So TE expression during aging, but also during other dynamic processes, is typically controlled via heterochromatin. So these regions are packed and sometimes also actively repressed via RNA interference. Now, when we age, there is literature that says that these regions become epigenetically open, and then specific transcription factors, like here, can bind to the loci of the transposable elements and then can be expressed. And it was found in several instances that they have an impact on the expression of genes that are closely located to these transposable elements that become activated. It has also been shown that when they are activated, they can cause what is called a sterile inflammation. So when transposable elements are activated, then the RNA is exported to the cytoplasm. And in the cytoplasm, there are specific pathways that recognize them. And these pathways then trigger an immune response, which then is potentially contributing to the so-called inflammaging, which is a process that we observe in older cells, that these older cells develop inflammatory signals that would lead the immune system to somehow, yeah, essentially attack them. And this may be an important hallmark of aging. And finally, the activation of transposable elements is associated with genome instability, so it could potentially also play a role in cancer. So what we are essentially asking is the genome-wide locus-specific quantification of transposable elements um, um, possible? Can we get algorithms that allow us to identify specifically which transposable elements are up and down regulated? So why this is a question, I try to answer in the next couple of slides. So when we talk about transposons, we talk essentially about two different types of transposons. The first class is the so-called class 1 retrotransposon class. This retrotransposon class, without going into too much detail, consists of so-called LTR elements, and among them are endogenous retroviruses and non-LTR retrotransposons. And we summarize line and sign elements like the L1 that I have talked about before under this subclass. And all of these transposable elements in class 1 use a copy and paste mechanism. That means they are transcribed from a certain locus, exported to the cytoplasm, and then they have certain proteins that allow a retro transcription and the reinsertion into the genome. So they are essentially copied and then reintegrated at some location back into the genome. So this is how they can spread. There is another class which uses a so-called cut and paste mechanism. And these elements are basically cut out from a region and then inserted into another region of the genome. Now, as you can see already, specifically for the class 1 retrotransposons, for active elements, at some point, assuming that the sequence does not change at all, it will be really hard at some point to find the locus where a certain retrotransposon originates from. Because if we do RNA sequencing analysis, for instance, or DNA sequencing analysis, and try to map the reads that come from these loci back into the reference genome, we may have a situation where the reads coming from this locus map to this locus as well as to this locus, and we can't really say whether only this or both or only this one has been active. But it becomes a little bit more clear maybe in the next couple of slides when I talk about TE families and how they arise specifically. So now assume here we have a toy genome with a transposable element here that starts jumping and reinserting itself. So obviously that's a class 1 retrotransposon that starts inserting itself back into the genome a couple of times. Now, as time goes on, these regions, fortunately for us, and we will see in a bit why, they acquire mutations, if you will, single nucleotide variants that would then allow us to distinguish these individual elements from each other, because only this here has these two mutations, and this one has these two, and this one has this one mutation. So they become at some point distinguishable because of the evolution that acts upon them. But remember, it gets a little bit more complicated. In some instances, the reinserted and mutated copies may still be able to jump. And in this case, we can see an element that has been mutated and is still able to jump, is reinserted into the genome and has acquired an additional single nucleotide variation. So as you can see, if we would be sequencing, let's say here, this genome, and or its transcriptome, maybe that's better. So if we would sequence the transcriptome of this individual here, we would not be able to know where the reads that we obtain from the transposable elements um, can map, because a read that comes from here could equally be mapped to this region, or a read from here could be mapped to this region, and that region. So it's hard to distinguish. But in a situation like this, it is a little bit easier. Now, nevertheless, what has been done so far is that these regions, these transposable elements here, have been grouped by sequence similarity into so-called TE families. And we can represent these TE families by essentially taking the consensus sequence, basically to reconstruct this original version from which they arose originally. All right, so this would be the consensus sequence, which is obviously not the same as this one, but hopefully close to the original one. And when we talk about Kimura distance, what we mean is how distant the consensus sequence is as compared to the copies from which we have derived this consensus sequence. So families where the distance between the individual elements and the consensus sequence is small, those are the sequences that did not have a lot of time or were under specific evolutionary pressures to develop mutations. So they have fewer mutations, meaning that they are potentially younger and have been integrated into the genomes more recently than the ones that have a larger Kimura distance. Um, just for those of you who are not so experienced with RNA sequencing, I keep that short. Mm, What we typically do, depending, of course, also on the sequencing strategy, we isolate RNA from the samples. Then for the Illumina strategy, fragment the fragments are extracted and converted into cDNA. The cDNA then gets adapters. Then the next generation sequencing is performed. And afterwards, we map these reads back to the transcriptome or the genome in order to identify the genes, quantify the genes that are expressed in the specific original sample. Now, one of the major points here is that the fragments that we obtain are short. But even if you use nanopore sequencing or other sequencing technologies that provide longer reads, in many instances, because transposable elements can be quite long, it is not possible to cover the entire transposable element. And specifically, it is often not possible to cover anything beyond the transposable elements. So, in essence, these short reads are in particular prone to be mapped to multiple locations and produce these so-called multi-mapping multi-mappers that make it really difficult for us to identify from which transposable element these RNAs were originally produced. And this is the reason, because these sequences are so similar, why previous research focused on the quantification at the family level, but not at the level of the individual loci. So let me quickly tell you how this works. So if you have here a transcript that is produced from this instance of a transposable element. Then you perform the NGS sequencing, and then let's say these reads come from this transposable elements. They need to be assigned to these transposable elements in the genome. And this is the problem because this assignment, as I said before, because of the multi-mapping reads, is sometimes not unique. So what is done typically is that these reads are aligned to these regions, and then all of these regions that belong to the same family, all of the reads that are mapped to the TEs that belong to the same family are then aggregated on this family level. And based on the reads that are aggregated on this family level, then previous papers and previous research, a quantification was made. So there are essentially two ways to do this. The TE-derived reads from RNA sequencing were either mapped to the individual loci of a family and then basically aggregated on the family level. This was a strategy that was used by TE tools and TE transcripts, for instance. These are two specific tools for doing this, while Salmon TE was generating a consensus sequence first from the individual members of a TE family, and then the reads were aligned to this consensus sequence. In any case, what you get are counts of reads that are aggregated at the family level. And here, because this comes up for the rest of the talk, it's really important to understand that difference, that we have individual TEs that share similarity. And in order to deal with the problem of this high similarity, we have worked previously on this TE family level. But we did not want to do this anymore and instead follow another approach. So what we did was we basically modified the tool slightly, very slightly, in order to be able to do a locus-specific analysis. So essentially, we were not doing this aggregation on the family level, but we were mapping the reads directly to the individual transposable elements here, and then quantifying the individual transposable elements expression also individually. And I will now basically show you a couple of slides how well or how bad that worked. I should mention that in the meantime, when we did this work, two tools became available that were specifically addressing the same problem, Telescope and Squire, saying that they would be able to quantify locus specifically. So how this algorithm works, you can find that here in this paper from Robert. And in order to test it, we were first selecting TE references containing 100,000 TEs. And based on these TEs, we were doing a read count simulation, basically to detect, to generate a ground truth. And we essentially injected then also um, um, a pure simulation, injected 5% of DETEs, or differentially expressed transposable elements, between two different conditions, because we wanted to find out first whether we can align the reads correctly, but then also we wanted to find out whether, if we compare two conditions with different transposable element expression, whether we would be able to identify the transposable elements that are truly differentially expressed. So this is what we call now the ground truth. So taking these simulated reads, we were applying these different tools here, the ones that we modified slightly, as well as the ones that were fit to do the locus-specific analysis right away. And then we compared these tools and tried to find out whether the locus-specific quantification is possible um, for TEs. And mm, what you can see here is that our modified version of the Salmon TE tool was performing surprisingly well, both for a setup using single end reads as well as for a setup using paired end reads. Now, if you're not familiar with these Illumina sequencing technologies, there is a specific mode where we do not only generate one read from an RNA fragment, but two reads, and this helps us a little bit also in terms of removing multi-mappers. It gives us a little bit more specificity. However, what you can see that it barely makes a difference for this top performing tool, this modified Salmon TE. So this expression detection worked well for all, and we were then specifically interested in the young elements, so the elements with the Kimura distance of smaller 5. And you can still see that Salmon TE worked best with the paired setup being yeah, substantially better. There were some tools that had quite, like Telescope, for instance, that had somewhat of a strange behavior with the setup. We didn't follow up on this. But in essence, what we were able to determine here is that the expression detection, at least, works already well with these modified tools. Well, expression detection is one thing. What we are always really after is to find out whether a transposable element is differentially expressed. Think about a young individual, a young sample, a young blood sample versus an old blood sample. We want to see what is different between these two samples, what is different between these two individuals. So differential TE expression analysis is a little bit more interesting than just saying, okay, that TE is expressed or not. So in order to do that, we were using DE-seq2. Not sure whether you have used the tool. It's a commonly used approach to detect differential expression of genes. Now we use this to also detect the differential expression of transposable elements. And we were, again, quite surprised about the pretty good performance. So let me quickly walk you through this plot for all of the TEs, so TEs of all ages. Here on the x-axis, you have the true positive rate, and here on the y-axis, you have the false discovery rate. The true positive rate tells you how many true positive how big the rate of true positives in among the differentially detected genes is. And here, this tells us something about how many falsely discovered differentially expressed transposable elements we have. So for a fixed FDR level of 0.1 here, that's the dashed line in the single end setup, we find that for this fixed FDR of 0.1, we still have a true positive rate of about 90%, which is quite well for the Salmon TE tool. And for the paired end setup, it gets a little bit better even. Now, but also for the young transposable elements, so the ones with a Kimura distance smaller 5, we still have with the modified Salmon TE tool, we still have quite good results in terms of true positive rate and false discovery rate. So we were essentially quite stunned by the fact that with only minor modifications of existing tools, it is possible to detect differentially expressed transposable elements. At least that's what our simulation suggests. So again, if you are interested to read this, this is the paper, and the following slides are essentially based on the groundwork here in this bioinformatics paper. Um, so we have to be interested, I have to say, in the biology of aging. And we were therefore applying this specifically to an age-specific context. So, of course, based on the simulation result, we selected salmon TE because the F score and the ETE detection works best. And we were sequencing 3 tissues of differently aged mice. Specifically, we were looking at brain, skin, blood tissues of 6 versus 24 months old mice. I've referred to the papers before, and most of the papers on that have analyzed TE expression, typically on the family level, have reported that there is a massive upregulation of transposable elements during aging. So the expectation that we had, well, not really, but the expectation one would have based on this data is that the epigenome, the heterochromatin that I've introduced before, is losing grip. The epigenome loses grip and fails to control the transposable element, fails to suppress the transposable elements during the aging process. And these transposable elements then get expressed, are exported to the cytoplasm, trigger all these nasty inflammation reaction, and these reactions then lead to the aging or the inflammaging of the individual. So TE being upregulated as a hallmark of aging, causing a lot of detrimental effects. Now, when we looked at the data here in brain, skin, and blood, specifically in the brain and the skin, what we found was that the magnitude of downregulation was at least as high as the magnitude of upregulation, meaning that the common narrative that TEs are generally upregulated during aging is not true. In fact, in the skin, we find a more downregulation than upregulation. So this sort of changes the narrative a bit. And of course, since we were able to pinpoint the individual elements of TEs that are expressed down or upregulated, we were also having a look at these. Now, what you can see here is the brain tissue for the 5 replicates of young versus the 5 replicates of old mouse brains. And what you can see is that we have a consistent upregulation here in this block, whereas in the old as compared to the young one. So a very clear picture that is essentially validated in each of these 5 replicates. Now, you can see that there is, as reported before, a substantial upregulation of these L1 elements here, the majority of them, but there are also a couple of instances where we have a downregulation in the old ones. And if you would be aggregating, let's say, the expression here on the L1 level, you would not be able to see that there are some L1 elements that are actually downregulated. And these may be, because they are the exception, particularly interesting cases. Moreover, because this L1 elements, if you remember the maternal care experiment from the Salk Institute I introduced before, particularly in this context, it may be really interesting to find out which elements specifically are up and down regulated. And this pattern is essentially repeated. Yes, we see a general upregulation of transposable elements here in these examples, but there are also elements where we have downregulation during aging. Now, this pattern is very similar in the other tissues. However, what you can also see is that the expression and the differential expression of transposable elements is also very tissue specific. So an element that is upregulated in brain may be downregulated in another tissue or not expressed in another tissue at all. So specifically here, what you can see is that in the skin, these endogenous retroviruses of type K are downregulated, whereas in mice, brain, they are upregulated. So a very interesting mosaicism again of this expression, which pointed a little bit towards a very tissue-specific function of these transposable elements in general. Now, this is not enough, and we further need to ask the question, how are these transposable elements regulated in general? How are they transcribed? And I'm going to introduce now two terms, which is dependent expression and independent expression. And I'll tell you in a bit what I mean by that. So when we look at transposable elements in the genome, what we typically find is that the transposable elements are often located in introns. And in this case, it sometimes happens that a gene is expressed here at its transcription start site, or short TSS, and then the primary transcript is produced for the entire gene locus, and later on, the introns are spliced out, which also means that the transposable element is expressed and may be detected if this intron sequence is not degraded, but it does not possess its own transcription start site. It is not regulated independently, but dependently based on its host gene. Whereas in the case of independence, we expect that the transposable element can be transcribed on its own, meaning that it possesses its own transcription start site, as well its own promoter, and its own transcription factors, really, that bind to this region and regulate its expression. And it was really important for us to distinguish TEs that piggyback, if you will, like in this case, on host genes as opposed to genes, transposable elements that are able to walk on their own. Mm, Without going into too much detail, there are specific protocols, sequencing protocols to do this. We use CageSeq. What you do is you essentially use the 5' cap of nascent, newly transcribed mRNAs to add a biotinylation or to add a biotin tag. And we can basically enrich these cap structures via the biotinylation and then pull these sequences that have just been transcribed out. Now, by doing this, what we will achieve is that we will have a pile up of reads that have this cap structure. And these reads that are piling up essentially at the same position will then tell us at which position there is a transcription start site. So at which position the polymerase has started transcribing. And we can not only find the transcription start site much better, but we also have a much better estimate on where the actual promoter is. So the region that triggers the expression of the respective transposable element in our case. But of course, this also works for genes. Now, with some bioinformatics, what you can do here, you find these cage peaks, and in these cage peaks, we can make a prediction on the well, nucleotide resolution start of the transposable elements. You will see in a bit that there's a little bit of a scatter around the transcription start site, but good enough for our case. Now you will see that. in a bit. So whenever we have a transposable element that has a Kh peak like this one, we consider it to be independently expressed. Now, when we look at the independently expressed transposable elements, again, what we see is a very large diversity and difference of the expression of TE families. So let's stay with the endogenous retroviruses here, ERV1, where you can see that independently expressed ERV1s are massively depleted as opposed to the background in brain. So basically a repression of these individually expressed transposable elements, whereas in skin, you've seen that a little bit already on the previous slide, whereas in skin, we have a strong enrichment of ERVs. So in essence, what you can see is that ERV1 expression is depleted in brain, but to some extent actively propulsed in the skin tissues. And we have done that for different families. Of course, again, interesting here, the L1 family in the brain seems to be, well, independently expressed L1s seem to be here enriched in the brain, very different from the other tissues. Now, when we look at the actual data in the genome browser, what we see is this picture, so that you have an idea how this data looks in particular. So here on the upper track, you can see the annotation as it is provided in the mouse from the mouse reference genome, UCC genome browser, whatever you use. With the annotations of a couple of endogenous retroviruses, ALU elements, again, B2 elements. So as you can see already, this looks quite messy, where individual elements appear to have jumped into each other, and then you have deletions of parts or insertions of parts. So it becomes quite messy. In the second track, what we have are the CAGE experiment results and the RNA-seq experiment results for the 6 months old mice. And here in the 24 months old mice, the same thing, CAGE-seq and RNA-seq. And these signals are basically the read tracks. So the higher the stronger the signal, the more reads have been mapped to this locus. Now, what you can see very clearly is that here we have a cage peak, which seems to be also active in the 6 months old mice. But here in the 24 months old mice, we see a pretty strong upregulation of this peak. But instead of having an upregulation only within this earth element, what you can really appreciate already is that this one individual upregulated start site leads to the transcription of the entire locus. So you really have what we later call a TE region or a TE island in which all of these elements are transcribed at the same time. So based on this result, what we were really interested knowing more whether TEs have arranged themselves in packs, packs of TEs that have sort of accumulated in a region. And one of these TEs, preferably the one that is at the 5' end, um, provides a transcription start site. So you can imagine that you have a TE and the other TEs are jumping into that region, basically piggybacking on the expression of the other TE to also be expressed. It's like, a queue where the TEs are queuing up and basically taking advantage of the regulation that is provided by the first TE. So to really make sure or to investigate whether that's the case, what we did was we first classified TE regions purely computationally without any without any experimental data, not to fall into logical traps here. So we took the annotation uh, and grouped the TEs, combined these TEs only based on distance and strand. So if two TEs were closer than 500 base pairs together on the same strand, we group them into a TE region. And in order to get a little bit more of a potential promoter region, we extended that TE region by 500 base pairs, and then we intersected it with our experimental data, specifically the RNA data, to find out about expressed TE regions. And then we further intersected it with our CAGE-seq data to get independent expressed TE regions and dependent expressed TE regions. So TEs are upregulated and downregulated during aging. They can have their own transcription start site, and they are expressed in blocks in TE regions, or what we now call it TE islands. So where does the expression start in the TE regions? Remember, we have not used any data other than the genome to define the TE regions or the expressed TE regions. It's purely computational. Now, here what you see are the starts and the ends of the TE regions that are expressed in skin and the blood. And what you see here is the density of the TSS signal. So very nicely, what you can see is that the TSS signal is highest right at the start of these TE regions. So it really seems that the TE regions have somehow managed to evolve in such a way that the first transposable element provides a transcription start site to all the following TEs. And this is true for the ones that are expressed in skin as well as in blood, with the highest value, the highest TSS peak here at the beginning. But when we looked at the brain, we found this, which is really awkward, and we were really troubled by that result, questioning our analysis, because here the transcription start site, well, there is an uptick of transcription start sites across all these regions here at the beginning, but the major peak is at the end, which would mean that they are essentially that there is a fraction of elements that is at the ends of these TE regions, and these are specifically only expressed to such an extent in brain. Now, this was particularly interesting, and I'll talk to you about this a little bit more in a bit. But before all of this, we wanted to know, because we have the TSSs, we also have the promoters, whether we can find specific transcription factors that may regulate the expression of transposable elements. And in fact, we were able to identify um, namely transcription factors of the SOX family that were significantly enriched in the promoters of the independently expressed TE regions. Now, specifically in the brain, what we did, we were interested whether these SOX5 or SOX family motifs in general have something to do with this peak that we observed. So what we did, we built a ratio of SOX5 motif occurrence and transcription start sites, and we plotted them across the relative position of the transposable elements. And what you can see is we have a very strong peak here at the end of this ratio of SOX and TSS, indicating that these peaks here may be particularly regulated. These transposable elements are particularly upregulated in brain through the activation of SOX, maybe SOX5 transcription factors. Now, in order to look into this a little bit closer, we were asking, do these SOX intersecting transposable elements are somewhat similar in other regards, in particular in length? So what we did, we took the TEs that intersect with a SOX transcription factor binding motif, and we simply plotted their length. And what you can appreciate, there is one peak that is very, very prominently around 1,000 nucleotides long. And it turns out that all of these elements that are so prominent here and causing this peak, this unusual peak at the end of our TE regions belong to the same TE subfamily, namely the L1 3' subfamily, which seems to be particularly expressed in the brain, but not in the other tissues. Now, of course, the question would be, what does it mean? But first, we then use these families to corroborate that they, in fact, have the Zox motif, a very conserved one, and they are apparently regulated through this motif. At least that's what we hypothesize now. Mm. So summing this up, many of the independently expressed TE regions are possibly regulated by the transcription factors of the SOX family during aging. It's not a good resolution, I'm sorry. Well, so what you see here on the x-axis and on the y-axis are log-fold changes. Now here on the x-axis, we have the log-fold change of the TE region, and here we have the log-fold change of the host gene in which the TE region is located. Because we wanted to know whether the upregulation of the TE region has a potential effect, or to be more precise, correlates with the changes of the gene. Remember, the TEs are now independently regulated, so they have their own transcription start site. But of course, it's a little bit of a problem to infer causality here. This is purely correlational because we don't know whether the gene causes the TE to be expressed or the TE causes the gene to be expressed. We can't say that for sure at the moment. But what we can say is that we have a group of genes in a cluster called PCDHB, so a protocaterin B cluster, that are upregulated in terms of the genes as well as the transposable element regions that are in vicinity to them. So this protocaterin cluster came up quite strongly in the brain region. What we also did, we did a GO-term analysis of the genes that are affected by this differential expression of TE regions. And what we found was a very substantial enrichment of synapse organization, dendrite development, neuron to neuron synapse, and glutamate receptor binding. So all of these hallmarks really point to neuronal functions, specifically to functions that are associated with the processes going on at the synapse. So neurons talking to each other, very interesting. Now, this enrichment becomes even more interesting because for every enrichment, what you really need to do is you need to propose a background. And in this case, what we did, we proposed the background of the neuron expressed genes. So this enrichment is on top of the genes that are already expressed in the neuron. So it is an even stronger signal as compared to the genes that are expressed in the neurons in the first place, which makes it quite striking because, in essence, what we believe to see here is a very strong enrichment of these processes um, in genes that are linked to transposable elements that are co-regulated, if you will, with transposable elements in their introns. So what we did essentially now was we were looking at the co-regulation again of this protocaterin cluster. And I quickly want to walk you through this mess. This protocaterin B cluster is a cluster of genes that has a beta cluster, is a product of a massive gene duplication itself. Now, the protocaterin cluster is known to be critical for synapse connections. So in this protocaterin cluster, we basically see what you have seen already in the Go term enrichment because, again, the protocaterin cluster is important for synapses and for the avoidance of self-recognition. Now here we have a couple of TE regions that were differentially expressed. And when we were looking at at all of the annotation items, both the protocaterin genes as well as the TE regions, what we find is that both the genes as well as the TE regions are upregulated during aging. And I want to now focus on this region here on this locus here with two TE regions just upstream of the protocaterin B15 gene. Again, the genome browser shot with the TE regions consisting of an L1 with a SOX motif and a transcription start site, and a second region consisting of 1, 2, 3 L1s and 1 B1 region with a transcription start site also in this region, this time also a little bit further downstream. Now, what you can see is during aging, both the first transcription start site is upregulated as well as the second one is upregulated. So essentially what we see is that these TEs are turned on. But more interestingly, maybe, is that the gene itself, the protocaterin B15, is downregulated, but still the expression here is higher. Now, what we have found in the meantime, and I have forgotten to put this data into, what we have found in the meantime is that actually from these transposable element regions, we are initiating transcription here at the transposable elements that runs through into the gene. So what we essentially have is upon this upregulation in the mouse brain, we produce fusion genes that contain both the L1 elements as well as the protocaterin B15. And this is somewhat very striking for us because these protocaterin genes have such an important function for neurons. And now we see this fusion or chimeras being expressed from transposable elements in the aging brains. That's certainly something we need to follow up on in the future. So what I can say is that independently expressed transposable elements could be involved in the tissue-specific regulation of genes. So coming to the conclusions, so genome-wide locus-specific analysis of transposable elements works surprisingly well. They can be up and down regulated. They have their own transcription start sites, and they are often expressed in blocks. In many cases, the first TE of a block, of a TE island, of a TE region provides a start site, and all of the others are piggybacking on this first 5' TE. They are potentially regulated by transcription factors of the SOX family, in particular in the brain, and they may be involved in the regulation of genes that have very tissue-specific functions. And yeah, so what I wanted to say is with these alternative alignment methods or modifications, um, they can help to get a completely new insight into an area of the genome of which very, very little is known. So I'm going to skip the P53 story because I don't have time. I'm talking too much. Variations of what I said before. And I want to thank these people. Yeah, Robert, this one here, was the one who was doing basically all the major work. So with this, yeah, I thank you very much. And if you have any questions, you can ask them now or drop me an email. And thanks a lot.
Thank you, Steve. Okay, now we have some questions here. You can also ask in Spanish. Steve understands very well Spanish.
Hi, thank you for the great talk. I have two questions. The first is that if the TEs you found in the brain could be uh, targets of some small RNAs, so other than the pig interacting RNAs that are found in germinal cells.
Sorry, can you ask the question again?
Yes. Uh, if the TEs you found in brain cells
Yes.
could be also targets of some
Ah.
other small RNAs other than piggy interacting RNAs?
A really interesting question. We have not looked into this yet, simply because the plethora of transposable elements that we have um, was so overwhelming that we had to first, or that we first decided to look into the genome regulatory actions of the TEs themselves. But from the data that we have at the moment, we have, yeah, I can't say anything about this, but of course that would be really interesting. What we have done specifically at the protocaterin locus, though, is we have tried to use RNAi to knock these transcripts down to see whether the knockdown of the individual transcripts exerts any particular effect on the gene expression. To this end, we have not found anything conclusive yet. So we can only say that they're upregulated and that they reach into the protocaterin, but knocking down so far was also a little bit problematic because, of course, we can only use, for this experiment, only use cell lines that express this protocaterin. We have not been able to dive into that deeper. But a very interesting question, obviously, but sorry, I can't give you anything.
Thank you.
We have one question from Jorge Manuel. Could you open your mic and your camera to ask the question, Jorge?
Yes. Hello. First of all, thank you very much for this talk. It was very interesting. And I have a question about the use of the Ts. Can Ts be used as biomarkers of some epigenetic modulations?
Um, um, I mean, I have to be careful here because I can't say for sure. But I think specifically because of the overwhelming tissue specificity, in particular because of this strong relation with aging, for instance. So you could think about, an epigen, well, in this case, a transposable element clock, using it as a biomarker to predict the chronological age. These things are being done. I know for sure. Yes. In my personal opinion, these transposable elements, at least in our hands, had such a strong tissue specificity that they are at least proxies, I believe, for the expression of um, of um, of their host genes, for instance. And using this knowledge, I believe that transposable elements can be very good biomarkers. Of course, the problem is that you really need to be sure that you are able, to measure the right copy. Because also in our data, in some instances, we can't be 100% sure that the reads that we are mapping to an individual locus are not misplaced. So this makes the whole business of using individual elements as a biomarker a little bit cumbersome. But I believe, yes.
Okay, thank you very much. Vielen Dank.
More questions? Okay.
I have two questions. So the first one is, have you used some sort of baseline when measuring these differential expression patterns? As in, are these changes with age in transposable elements, expression changes in age with age, more significant compared to other genomic elements that have been related with age?
So, I mean, in I mean, to sort of, yeah. So maybe I answer this question in terms of logfold changes, because significance is reached or is not reached. It's a binary decision. For me, it's always a little bit difficult to explain that. But if we are talking about logfold changes, uh, what I can say is that there are many genes that have a stronger differential expression than the logfold changes of transposable elements that we observe. So in general, I think it's fair to say that also because we can't measure all the transposable element loci, the logfold changes for the genes, at least from what we see, are typically much stronger than the ones that we observe in transposable elements. Of course, it depends on the individual locus, on the individual gene, but the, let's say, dynamic range, if you will, of genes is still larger than the one of transposable elements. Now, having said that, this has also something to do with the fact that we, of course, for the mapping strategies, we are, with the Salmon TE tool, we are in many instances not able to confidently assign the reads to an individual locus. So what we do is we lose a little bit of signal at individual positions of the transposable element. So in genes that are unique, where across the entire gene, the sequence has a high uniqueness, what we can do is we can map the reads consistently across all of the exons. But in transposable elements, what we see is that there are regions where we have a loss of signal. So in essence, what we are losing here is depth, and this may also play a role why the dynamic range or the log fold changes that we observe in the few tissues and in the few conditions, I have to say, that we looked at the moment are smaller. But I expect that to be the case also for other tissues and other conditions.
Okay. And the second question is, how would one work with transposable elements with organisms that don't have a reference genome?
Yeah,
How would one go about that?
yeah. Well, I mean, the locus specific analysis is obviously also a problem in genomes where we have reference genomes. So think about L1 elements in mice that are still active. So there are some L1 elements that are still jumping around. So let's say one black 6 strain from Jena may be different from black 6 strain that you just obtained from Jackson Laboratories or from Janvier, simply because these L1 elements are active. The actual genome of, although they are both black 6, of these individuals look very different. So you can never rule out that specifically for actively retrotransposing TEs, that your reference genome is not accurate. And this is true for mice. So it's, because, yeah, true for all of the elements that are still active. What can I recommend? So the only way that I can see really is to take the species, the organism that is most closely related, and try to find out whether you still have an opportunity to map locus specifically. But I can say already, imagining the evil reviewer that will read your work, you may have problems explaining this. In particular, when you deal with transposable elements that are very active, and in particular, if you deal with species that have no close relative. So, I mean, essentially, what you can also do is you can go to the databases that contain the different classes, families, subfamilies of transposable elements, and map your reads to these consensus to get an idea which specific family, for instance, may be expressed. But yeah, I consider this as a big problem, and this type of research really hinges on good reference genomes.
Okay. Thank you very much.
Sure.
Another question?
Hello. Thank you for your talk. One question that I have been asking is, do you think this expression pattern could change if the tissue is under certain stress, like maybe a disease?
Yeah. Actually, that was a little bit what I wanted to show in the second part, but because I talked too much, I wasn't able to present this. So what we did in our lab, that was Martin and Robert, we have a simple we have taken human cell cultures, MCF7 cells, for instance, and activated them or treated them with nutlin, which is a small molecule that activates p53. guardian of the genome. You may know this, a gene that is typically upregulated in response to DNA damage, for instance, and a major transcription factor that essentially can drive cells into apoptosis, plays a role in senescence and so forth. Very powerful transcription factor. Now, the current literature says that p53 is a transcription factor that suppresses transposable elements. And what we found, in fact, is quite the opposite. We found that p53 activates many transposable elements. Now, what I can say from these experiments is that environmental changes, radiation, for instance, um, chemicals that would activate p53, but I'm sure also other important transcription factors would also alter the transcriptional landscapes of transposable elements. Now, for the p53, it was in particular very interesting for us because the field at the moment believes that there is also regulation, but in the other direction. It's believed that p53 downregulates transposable element expression. And in fact, we find that we have a quite substantial upregulation. So yes, I'm sorry. The answer is yes. I'm sure. I'm certain. And the p53 is one important piece of evidence.
Thank you.
Hello, thank you for your talk. This is more a comment. I don't know if it can be useful, a comment related to the software. You show that Salmon T has a good performance for the locus-specific mapping of transcriptional elements. And as far as I understood, the problem was the accumulation of mutations in transcriptional elements for the mapping. The problem is multi-mapping. And I immediately thought of the HLA genes, which are highly polymorphic genes related to the immune system. And maybe it is useful, but this is more for genotyping, but there are softwares specialized to manage these highly polymorphic regions. So this might be, the concept might be different, but maybe the algorithm could be useful for making a more sensitive prediction of where the locus is.
Yeah, it's a super, super cool idea. Thank you very much. So in many of these tracks, just as a side note, in many of these tracks, what you've seen are these expression signals, and these have not been obtained with this Salmon TE quasi mapping tool because it just aggregates counts over annotation items, but it doesn't exactly tell you where the gene is aligned. So it's a pseudo alignment, not a real alignment. So in order to get these tracks, we needed to realign the reads with conventional methods. So real dynamic programming. And like also from there, we saw that for many of the transposable elements, the alignment rates as well as the expression levels that we get is not as good as the Salmon TE, but quite similar. Yeah, so HLH, that's a very good idea. Something we really would want to test, I think. But one of the I think one of the key messages for this transposable element is that um, it is quite strange that so far no one has really attempted to use conventional alignment methods and benchmark them, um, because already with the simpler, straightforward method, what we see is that there is a pretty good resolution already. So I don't know why people have refrained from just doing it. But yeah, sometimes, right, there is no answer.
Okay. Thank you very much, Steve, for this super enlightening talk. Unfortunately, he's not going to be around, but maybe you can write an email to him if you have more questions or would like to ask for opportunities in Germany, in his lab. And thank you, and see you around.
Thanks a lot. Sorry not to be with you. And I'm hungry. I want alambre. Anyway, Maribel, maybe you can send me one over. Like here also, it's super hard to get corn flour, like the Mexican one, and super hard to get all the ingredients. So maybe you can send me a box with flour so I can make my own tortillas again.
Tacos al pastor.
Tacos al pastor, por
Sí.
ejemplo.
Okay. Bueno. Okay, chao.
Chao.
Chao.